Hello everyone, welcome to the March Didsbury Sidebar. Our speaker tonight is from Manchester University's School of Biological Sciences, I think. It's Professor Ray O'Keefe, and he's going to teach us all about splicing. Right. Thank you, Ray.
Okay, hello everybody. Thank you so much for inviting me, Sarah. And it's really nice to, I guess, see everybody's names virtually here via Zoom. And it's a shame we can't be together having a beer either before or after my talk. Let me share my screen and I can start. Let's see. Okay, that's good. All right, let's get this going. Get a pointer out. Okay, great. Title, Splice Up Your Life, RNA Splicing in Health and Disease. So what I'm going to talk to you about tonight is what RNA splicing is, what diseases are caused by defects in splicing, how we study diseases caused by splicing defects, and how we can treat diseases caused by defects in splicing. Now, I guess to understand what splicing is, we need to understand what RNA is. And I'm not sure if everybody knows what RNA is, so I'll give a bit of background about what RNA is. And sort of in the basic level, RNA is a messenger. So RNA carries the information from the DNA to make proteins in a cell. So as you can see here, you've got DNA. DNA then goes through this process of transcription to make an RNA. And the information from the DNA can't go directly to make a protein. It has to go through this intermediate called RNA. And then this RNA, it goes through a process called translation. And then that goes on to make a protein. And this process of DNA making RNA, making protein is this so-called molecular biology central dogma, which was first proposed by Watson and Crick soon after they discovered the structure of DNA. Now, if you look here, the DNA is making an RNA that is similar size and similar length to the DNA. And the key thing here is this RNA that is the message, or the messenger RNA or mRNA that carries the message. This RNA is an exact copy of the DNA, but we don't need all the information that is in the RNA to make the protein. So we only need a very small part of this RNA to actually go on and be translated to make protein. And that's what RNA splicing is. And this sort of RNA splicing, or the idea that not all the information in the DNA is required to make the final message to make proteins, came about sort of in the 70s, in early experiments in the 70s, when scientists were looking at viral DNA and viral messenger RNA and looking at them and comparing them with each other. And you can see here from this electron microscopic image that what they've done is they've hybridized this messenger RNA to the DNA from a virus. And what you can see is that the mRNA, the final mRNA that goes on to make the protein, is a lot shorter than the DNA, the viral DNA. And there are these big loops that come out. So A, B, C, these big loops come out, and they don't correspond to any of this information in the messenger RNA. So this idea that not all the information in the DNA is required for the message, and because you have these loops, what that means that genes that code for proteins and make the messenger RNAs, they're split genes, and there has to be a way for this RNA, or that's first made from the DNA, to then be cut down and spliced into this shorter messenger RNA that corresponds to what you need to use to make the protein. And this idea was discovered by these two guys here, Rich Roberts and Phil Sharp. They did some of these experiments and other experiments to show that our genes, and genes in viruses as well, are split, and that the RNA needs to be processed or spliced to get this final messenger RNA to go on and make a protein in the cell. And this is really important because proteins are the essential sort of catalyst of a lot of things within our cells. And they won the Nobel Prize in 1993 for this discovery. Okay, so not all the information in the DNA is required to make the final messenger RNA, and that means that the genes are split and they have to be, the RNA has to be cut up and put back together in some way, and that's the RNA splicing. So I'll go into a bit more detail of how the splicing works. So you have your DNA here, and what we can define are the bits of the DNA and the bits of the RNA that are important, that contain the information that we need to make the proteins. And these are defined, the bits of DNA that code and make the RNA that then will go on to make the protein. These are called exons. So exons contain the important information to make the proteins. So here you see there's 5 different exons. And those exons are interrupted by things called introns. So bits of DNA or bits of RNA that are called introns. And the introns and the information in the introns aren't needed to make the proteins. So these introns here, one there, and one there, all these introns need to be removed. And they need to be removed precisely so that the information that is found in each exon comes together correctly to make the correct protein within the cell. So you've got these exons, which are the bits of information that are used to make the proteins, but in between these exons are these things called introns that aren't needed to make the protein, and they need to be removed. So they need to be spliced out. So if we go on to the next slide, what I've done here is these are sort of some typical RNAs that you can get after splicing. So what happens here, as you can see in this first example here, each of these introns, the 1, 2, 3, 4 introns between 5 exons have been removed, and these 5 exons have been spliced together. Now, what I've done here is I've put underneath each of these exons a bit of information, because that's what the exons are. They are the information to make the proteins within the cell. And I've just put a couple words here that make a bit of sense. So protein for heart, eye, and kidney. So that in my sort of imagination here, I'm thinking this protein will be used for the heart, eye, and kidney. Now you can see here that that's in some situations you get, if you've got 5 exons in this gene that get put into the RNA gets spliced, and all 5 of these exons can be put into the messenger RNA or the mRNA. But in some situations, you can get some of these exons either being skipped or swapped around. So in this case, we only get 1, 2, 4, and 5 being put together. And then this protein will be for just the eye and the kidney and not the heart. So this protein is slightly different because it's removed this bit of information. And the RNA that makes this protein will make a protein that's slightly different because it doesn't have this bit of information in it. The same thing here, we've got exons 1, 2, 3, and 5 coming together, and we're skipping exon 4. So this protein will only say be in the heart and the kidney. And this sort of excluding or swapping around of exons is called alternative splicing. So some of these RNAs can be spliced alternatively. So if you have a gene, you can get all the exons being included like you see here, or you could get some variation on those exons being joined together, and you can get different functions for these RNAs, and they make different proteins. So some RNAs can be spliced alternatively. And this next example is a really interesting example that I always love showing the students. This is an extreme example of this type of alternative splicing. And this is this gene called a DSCAM gene, which is required for correct neuronal guidance and self-avoidance. So you can see here, you've got nice neurons here, but when you get rid of the DSCAM, they get disorganized, they start associating with each other. So this DSCAM gene is very important. Now, if you look at this structure of this gene here, it's really complicated. it has 24 different exons. So 1 to 24 here, and then the introns aren't even shown because there are these little lines in between here. But the interesting thing here is there are these 4 exons. So exon 4, exon 6, exon 9, and exon 17. These exons can be spliced alternatively. So every time you get a splicing, you only get 1 exon 4 in, 1 exon 6, 1 exon 9, and 1 exon 17. But say for exon 9, there are 33 alternatives that you could put into this position by this alternative splicing. So if you can imagine, if you do the math, if you're really quick at the math, what you can potentially have by having all the different combinations of these alternative exons in a messenger RNA, you potentially get 38,016 different proteins made just from this one gene. So this alternative splicing is really important. You can see here, you can get very, very large diversity of proteins from a single gene, or the information within a single gene. And that's really important in a lot of processes within our cells. So alternative splicing is really a key process for organismal development and cell differentiation, and for things like guiding neurons and having these neurons stay apart instead of coming together. So this alternative splicing is really, can become really complex, and that's because in some complex organisms you need this complexity to give you the diversity that you see. So that sort of gets onto the question, why has RNA splicing evolved? Why do you have this really complicated mechanism here to get all these different proteins? Why don't you just have 38,016 different genes to make an individual protein? That's one of the questions, a good question. And the reason why is that if you had all that many genes to make those proteins, you'd have to have a huge, huge amount of DNA and a huge genome. So splicing has evolved. Splicing occurs in everything from viruses up to humans. And splicing's evolved to increase the informational diversity and functional capacity of genomes. So for example, in humans, we have approximately 21,000 genes. And these genes can make almost 1 million different proteins by using RNA splicing. So we have this number of genes is actually very, very similar to other very simple organisms. So there's one example that people usually give, and there's a model organism that we use called C. elegans, which is a very small nematode, a very small worm type organism that contains almost just up to about 400 cells. It also contains 21,000 genes, but it doesn't code for as many proteins. So having this ability to take these genes and to get a wide variety of different proteins with slightly different functions has really been important for the development of more complex organisms like ourselves. So the RNA splicing is really important for getting this protein diversity that organisms need to get more complex, really, without expanding and having a huge genome to carry all those genes. Okay, so what is it that actually carries out this splicing? So what actually removes the introns and puts and splices the exons together? Now here, what I've got here, or what's shown here, is there's an exon, there's an intron, and an exon. This is a very simple sort of gene here with just 2 exons and an intron. Now the intron needs to be removed precisely. You have to have a machinery that cuts exactly here and exactly there to remove that intron and put the 2 exons together so the information that they have makes sense. Any mistake in that is going to be detrimental. And the machinery that does that is this machinery called the spliceosome. And the spliceosome is a complex that you find within the nucleus of the cell, And when the messenger RNA is made with an intron, it comes in and recognizes where the exon sequences are and the intron sequences are, usually by looking at this very specific sequences here at the junctions between the introns and the exons. And this spliceosome complex comes in, and you can see here it's interacting with the exon and the intron. And what it does is it finds where the ends of the intron are. It cuts those two ends, and then it joins the two exons together, releasing that intron that it doesn't need to make for the mature messenger RNA. And this is quite a big complex. We know quite a bit about this complex, how it comes in, recognizes the sequences in the RNA to know where the intron is and where the exons are. And it actually cuts out the intron and splices together the two exons together. And it's very similar to how, say, a film editor edits a film for after production, and the film editor wants those bits of film to come together the right order and in the right sequence and to make sense. So the spliceosome is doing the same thing. It's taking out these introns and putting the exons together so they make sense when they're together to make the right protein. And if you can imagine that this spliceosome needs to be very efficient and very good at recognizing where the exons and introns are and putting them together properly. So if we go back here, it's got to cope with something like this, very complex, and knowing when and where to put these alternatives into the messenger RNA. Okay, so you've got the messenger RNA that contains introns, this pre-messenger RNA that contains introns. This cellular machinery called the spliceosome comes in, recognizes where the introns and the exons are, and removes the introns and puts the exons together precisely to give you a mature functional messenger RNA that can go on and make a protein. And it's amazing how this happens, and it doesn't go wrong all the time, but it works, it's really good, but in some situations, it does go wrong. And that's where sort of RNA splicing and disease comes in. So defects in splicing are actually a significant cause of disease. And now we could estimate that about 30 to 40% of all genetic mutations that are in the human population that come up or that are passed on between some people, 30 to 40% of those genetic mutations influence splicing. So it's a real significant cause of disease. And you can imagine that there's a lot of ways that this splicing could go wrong. So you could get mutations in this pre-messenger RNA, making mutations maybe near these splice sites that'll prevent the spliceosome from recognizing where an intron or an exon is, or having mutations that either keep an intron in, don't get it, make it go out, or put the wrong exons together. So you can get mutations that are in the RNA, but you can also get mutations that are in the spliceosome that affect the function of the spliceosome. So mutations that affect this spliceosome function. So there's two ways. So that's why there's such a high percentage of mutations that can influence splicing. So you can have mutations that are in the so-called substrate that the spliceosome has to come in and splice, but you can also have mutations in the machinery that carries out splicing as well. And that those mutations can affect how efficiently or how accurately the spliceosome removes these introns from the messenger RNA. So RNA splicing is a significant cause, or when you have defects in RNA splicing, it's a significant cause of disease. Now, some diseases, so that again, I've talked about these sort of two places where you could get the mutations. You can either get the mutations in this pre-messenger RNA or in the spliceosome. So there are certain classes of diseases that you can get that are either from mutations that you get that affect the mRNA. So some diseases caused by mutations in the mRNA that change splicing. So some examples are cystic fibrosis, muscular dystrophies, hemophilia, progeria, which is premature aging, breast cancer. There's quite a few mutations that are associated with splicing that cause breast cancer, neurofibromatosis type 1, and spinal muscular atrophy. So those are when you get changes in this mRNA that will then change, mutations in the mRNA that will then change and cause splicing to be defective. But then, as I said, there's also some disease caused by mutations in the spliceosome. So if you get mutations within the spliceosome, it may not be able to splice messages properly or the right way. And some of the diseases that are caused by mutations in the spliceosome include some cancers, some types of dwarfism, retinitis pigmentosa, which is an eye disease. I'm not even going to try to say that, male infertility, and a hair loss disease as well. So some of these have bad names. That's why I'm not telling you the names of these 4 craniofacial disorders that are also caused by mutations in the spliceosome. So you get these 2 types of mutations. You get mutations in the RNA that needs to be spliced, and you can get mutations in the spliceosome, which is actually the machinery that's needed to splice these messenger RNAs, remove the introns, and join the exons together. And our research has really been interested in looking at these mutations in the spliceosome. And we've spent a lot of time over the past sort of 5-10 years looking at these craniofacial disorders, which are caused by mutations in parts of the spliceosome. So I'm going to talk to you a bit about the work that we're doing now to look at these craniofacial disorders. But one of the sort of interesting questions when we talk about these mutations in the spliceosome, so if we look back here, the spliceosome is required for the splicing of every single messenger RNA in the cell. You can't get splicing without the spliceosome. So if you had talked to me, say, about 10, 15 years ago and told me that, oh, if you get mutation in the spliceosome, you're going to get disease, I would have said, well, I think you're crazy because the spliceosome is essential. If you mutate the spliceosome, you wouldn't, splicing is required for every messenger RNA in every cell in the body. If you mess up splicing, you're probably not going to be alive. Nothing's going to, it's going to be bad. It's going to be really bad. But what we found over the years now is that there are certain types of mutations that you get within the spliceosome that affect proteins in the spliceosome that can give these very specific diseases that you see here. So you get mutations in the spliceosome, but they only give you very, very specific types of diseases. They don't affect all the cells in the body, and the cells are still alive and people are still alive with these mutations. So a real big question we've been trying to answer over the past few years is why do the mutations in the spliceosome, which are required splicing of all RNAs, cause very specific diseases? Why doesn't it affect everything? So one of the craniofacial disorders that we've been looking at most recently is this craniofacial disorder called Burn-McEwen syndrome. And it's quite a subtle genetic disorder, and it's quite rare as well. And the clinical features are that the patients usually have external ear abnormalities. They may have some hearing loss, cleft palates, and an underdeveloped jaw, so a jaw that's gone in a bit, lower eyelid defects or no lower eyelids at all. And they also have this thing called choanal atresia, which I'll get onto what that is in a minute. And that's quite serious. But the genetics behind Burn-McEwen syndrome is that the gene mutations cause a reduced amount of one specific spliceosome protein. So it doesn't get rid of it. It's still there, but there's just a very lower amount, a reduced amount. And we think having that reduced amount is one of the main reasons why we get this very specific order and not everything isn't affected. So these are the clinical features, and the choanal atresia is quite serious. So what that is, it's a blockage in this passage between the nasal cavity and the pharynx. So here you can see there's obviously two passages there from your nose down into your throat. In this case, one's blocked and one's open. But in these people with Burn-McEwen syndrome, most of the time, both of these are blocked. And it's very serious for newborn babies because, as probably some of you know, newborn babies are obligate nose breathers because they're suckling all the time and they need to breathe through their nose. And if a baby's born with this, it's very serious and it requires immediate surgical intervention. So actually, one of the things that we've done doing the genetic analysis of the Burn-McEwen syndrome patients is we can at least give genetic counseling to families where this runs in the family. So if they're having a baby, they'll know whether they have the Burn-McEwen syndrome, and they may have the possibility of having this choanal atresia. So when the child is born, there'll be surgeons there ready to act and to get in there and give that surgical intervention and open this up, which isn't that bad of a procedure to do, but it gets the opening open and allows the baby to breathe.
Hi, Ray. Well, we've got a couple of questions in the chat.
Oh, sure. Sorry,
No, it's all right. It's all right. I don't like to interview really, but yes. Sue Foy asks, are there any of these mutations beneficial?
Are any mutations beneficial? You can get definitely beneficial mutations that make splicing work better, or so yeah, so okay, any of the mutations beneficial? Yeah, so there is a possibility. So during evolution, you could get mutations that then become beneficial, that maybe affect splicing in a way that gives a protein a new function. So yes, definitely.
And yeah, okay. And Carolyn Jones asks, is there only one spliceosome per cell?
No, there's thousands of spliceosomes per cell. Yeah, so yeah, and splicing acts really quickly. So the cell has thousands of spliceosomes that are splicing the RNA as it comes out from transcription from the DNA, and you have loads and loads of these spliceosomes coming in and cutting out the introns and splicing together the exons. Yeah, that's a good question. Okay.
Okay, thank you.
Okay, would CF both parents have to be carriers? So where has the splicing gone wrong? Okay, so yeah, so the splicing's gone wrong because you get a mutation. It's sort of similar to what I'm describing here, that causes splicing that gives reduced function of the protein, but not completely misfunction of the protein. So it's sort of different levels. So with CF, what you get is you get a mutation in a splice site that then affects the message that you get, but you still get a message, but then that message makes a protein that's got slightly different function than the normal CFTR protein. Will a mutation in the spliceosome affect them all? Okay. I'll get onto that. That's sort of what I'm getting onto now, what I'm talking about. But it depends on what the mutation is in the spliceosome. If it's a major mutation in the spliceosome, it'll affect everything and you won't have any cell viability, you won't have anything. But mutations in spliceosomes, they're generally just bringing down the function of the splicing a bit. And as I'll get onto, they only seem to affect certain messages. Let me just stop my share there and go on to the questions because my PowerPoint's not going forward anymore. Okay, would a mutation is it known why the introns are there? Do they have or had a purpose? That's a really good question, Ian. So introns have evolved over time to give us this protein diversity. There are some functions for introns. So some introns actually code for RNAs that are functional on their own. So yes, some introns have purposes and they could code for things, but really what's happening with the introns is they've been introduced to try to during evolution to give this diversity by splicing. But there are some introns that when they're removed, the RNA usually is degraded, but in some situations, those RNAs go on and function as RNAs on their own. Okay, let's see if I can get my presentation back up and go back if there's no more questions. Those are really good questions. Right, so let me get back to sharing. Right, get up to where I was. I'll keep a better eye on that chat when I get there, when I get going again. Okay, great. So that was a cranial atresia. So the thing about these craniofacial disorders is that the craniofacial features form early in human development. And what happens is you can see here, this is sort of development of the head and the face in humans. What happens here is there are these certain cells called neural crest cells that start in the early embryo, and they migrate and to certain positions, and you can see that they're starting here and they already got sort of their fate. So these purple ones will then go on to make sort of the higher part of the head here and the eyes, and these blue ones more in the lower jaw and the nose area. So these neural crest cells come along, they go and they migrate to these different areas, and then when they get to those different areas, they start differentiating and forming these craniofacial structures that you find in a fully developed head. And it's the migration of these neural crest cells that are essential for this proper craniofacial development. Now, so we think when splicing is going wrong, it's going wrong in these early stages here. Something's happening to these neural crest cells that's making them either not migrate at the right time, migrate to the wrong place, or not migrate away when they're supposed to, like with the cranial atresia, that the cells are supposed to come apart and form that tube, and they don't do it. So it's all happening here early on. Unfortunately, as a scientist, we can't look at this. We can't look at early human development and look and see what's going on here. So we've had to devise some different methods to look at what's going on here. And the way we've done this is we've gone to some of these new stem cell technologies that we have these days. And it's amazing. What we can do now that we couldn't do, say, 10, 15 years ago is amazing. So over that time, what we've been able to discover, scientists have been able to discover, is that we can make these certain stem cells from a patient's blood. So what we do is we get a patient, a Burn McEwen patient. All we need to do is to take out their blood, which is a very simple procedure. People aren't too bothered about that. And what we can do is we can take these blood cells, and what we do is we reprogram them in a petri dish in the lab into these stem cells called pluripotent stem cells. And what pluripotent means is that it means that these stem cells can then go on and develop into any other cell type. So we've induced these blood cells to go back into their sort of primordial state, their initial state, where they can now differentiate and go off and be any other cell type. So this is really neat. It's not easy. It takes a while, and it's quite a complex procedure to do, to reprogram these blood cells into these pluripotent stem cells. But when we do get these pluripotent stem cells, they have the exact genetic background of the patient that we're interested in. And then in our situation, what we then do is we take these stem cells and we differentiate them into the cell type that we're interested in. And in this case, it's these neural crest cells, which are the cells that go and migrate and make all these craniofacial features eventually. So these pluripotent stem cells are good to use, hard to make, good to use, and we can differentiate them into the cell type that we want. So we can differentiate them into neural crest cells and ask, what do the neural crest cells, or what are they like? What's happening with the splicing in cells from a patient compared to, say, one of their relatives, mother or father, who doesn't have the disease? And what happens to splicing in these cells? And what we found is that when we have this mutation in the spliceosome that brings down the level of this one splicing protein just a bit, but not too much, is that there's only a small set of RNAs that are spliced, that are not spliced correctly in the patients. So compared to the parents, there's a small set of RNAs that are not spliced correctly, and these RNAs are, we found, required for these neural crest cells to migrate and differentiate. So what happens with these neural crest cells is they start off like this, where they have cell-cell contacts, and then they change their splicing program to become these migratory cells that then go on to the different parts of the head and then differentiate into the different cell types and to the different structures. So you can see here that there's a number of different genes that have different splicing here. So this is the example here that we actually found. So in these cells over here and over here, you get this splicing where you get inclusion of this exon 4. But when you get the cells that start migrating and moving around and they change their structure, you don't have exon 4 in there. And one of the genes that we found that is in this small subset is this one gene here called TCF7L2, where what we see in the patients is that they still have, most of the time, that in the patient cells, when we make them into neural crest cells, they haven't removed this exon 4, and they still, and they haven't gone to this form here. They're still in this form, so the neural crest cells are still in this sort of stage here, and they haven't gone as well into this migratory stage. So I did see that some questions came up here. Let's just see. If I can see it. I see the chat. Gonna have to stop and see the chat. There we go. Okay, right. So Paul Fletcher, thanks. Is splicing the reason why a single point mutation can cause major changes in the body plan, such as conversion of a fin to a fully formed limb? Wow. Okay. It's possible, yeah. You can get a single point mutation that would, so what happens when you get a single point mutation that would affect splicing, the effect can be quite drastic. So you can either include or exclude a whole exon, and that could change how the body plan was formed. So it could change an important protein in the pathway for making a fully formed fin and make it form a limb. So there are experiments where people do this in Drosophila, in the fruit fly, and they can change the splicing, and instead of having a wing coming off one position, it's got a leg coming off where the wing's supposed to be. So yeah, you can get changes in splicing that give very drastic changes like that. I have a related question about tRNA, specifically tsRNA, but appreciate this topic beyond this talk. Does the speaker here? Okay, I'll maybe come on to that later. Anything with tRNA. So let me get back onto my slides. Share screen. Great, so, okay, where was I? Right, so that was, I've taken a bit longer than I thought. So let's see, so that was the end of the cranial facial stuff. So Yeah, so with the Burn-McEwen syndrome, I've talked on a bit too much here, but the reduced amount of a spliceosome protein causes incorrect splicing of only a specific subset of RNAs. And that leads to these cranial facial defects. So I've got a bit more. So I haven't really talked about treatment. So obviously we haven't gotten to the point where we could do treatments for Burn-McEwen syndrome. We can't get into and treat the embryos during the early development, but maybe in the future there could be some ways we could do that. And knowing the information that we know now could be useful for that. But there are some really good examples of how we can treat diseases caused by defects in splicing. And I'll go through this example quickly because I know of time's come on a bit. So with spinal muscular atrophy, there's some really good therapy for spinal muscular atrophy. And with spinal muscular atrophy, what happens is that you get a mutation in a deletion in a gene called SMN1. And you get, so that gene isn't there anymore, but we have in our body another identical gene or almost identical gene called SMN2. And that gene only works 20% as good as the SMN1. Now I'll go through this quickly, but the difference between SMN1 and SMN2 is only one bit of the DNA. So there's a C here and a T there. But what this T does is it makes gives only 20% normal protein with the exon 7 in here. And when you get this T change, what happens is this exon 7 is now skipped. So what researchers are trying to do, and I've done, is think of ways, how can you increase the amount of exon 7 included in this SMN2 to make more of this normal SMN protein. Now, this story is a really great story from about this drug called Spinraza or nusinersen, and it's an antisense DNA oligonucleotide drug. And it was discovered and worked together by these two scientists, Adrian Craner, who is a colleague of mine back in America, and this other guy, Frank Bennett. So Adrian works at Cold Spring Harbor Labs, and Frank Bennett works for this company called Ionis, which develops these antisense DNA oligonucleotides. Now, summarizing a whole bunch of work, what we know is that there's a the antisense oligonucleotide can come into the cell and it binds to right near this exon 7, preventing a protein from binding and allowing the inclusion of this exon 7. And this is like a summary of about 15 years of basic research. And with the clinical trials, these were really good. So in phase 1, using this Spinraza, you could get improvement in all of these criteria for the SMN patients. So phase 1 worked really well. Phase 2 worked really well as well, looking at infants with SMA, whereas before this was older patients with SMA. And then in phase 3, when they went and did a randomized, double-blind, completely controlled experiment, what they saw is that the patients who got this DNA oligonucleotide to change splicing had greatly improved ability to move and met all the milestones way before they were supposed to having SMA. And that's compared to the controls. And it worked so well that the trial was ended early. All the patients that had the control that weren't getting the drugs got the drug. And this drug was improved really quickly by priority review. And it was approved by the FDA in America 2016, in the EU in 2017. And it was the first approved treatment for SMA. Now, I know I've gone on a bit, but I just want to show you this quick video. This is probably the last thing here that I'll show you of really the effect that this has on the patient. So. No, it's not going to do it. Right. Stop sharing. Sorry. Right. Sorry about that. Let me get to all these all these Zoom things. I got to try to do it once. So let's get back onto this. And okay, if I go into there, there we go. That should work now. Right.
And the point.
Oh.
And the point.
Emma, do you want to say hi?
No.
Emma, she is a 3.5-year-old, sassy, smart, fun little girl.
Emma was diagnosed with SMA, spinal muscular atrophy. It's self-explanatory, muscular atrophy. These muscles are starving for the protein from these motor neurons, and her little body doesn't produce that protein or an adequate amount.
They said that she would never stand, never walk. It would eventually affect her upper body and her breathing. It's very scary enrolling your child into a clinical trial. We don't know what the effects are going to be long term. We still don't, but at this point we knew if we didn't do anything what the outcome would be. She was diagnosed at 1.5 years, and at that point they said that she couldn't enter into phase 3 until she was 2 years old. In that 6-month period, she went through a lot of regression. It got to the point where she couldn't even sit up and hold a bottle without tipping over. Her diagnosis was pretty grim. Sorry. Okay. Her lifespan would be shortened, and basically we'd watch this disease progress over time. On the day of her 2nd birthday, we literally drove to Colombia to get her screened for the Ionis nusinersen trial. Then 3 weeks later, she got her first injection, and we started noticing changes in her around the 2-month mark. She crawled all the way from the living room to our master bedroom, and we were just in shock. She now stands. We have little orthotics that help her now with a walker, and we just started her with crutches. Not only that, the disease has stopped progression. She's no longer lost anything. In fact, every time she goes in for another injection, she gets a little stronger. As soon as we found out that the drug trial that we were interested in was actually discovered here at Cold Spring Harbor Labs, we had it come down. For me, Dr. Craner is a superhero in regular clothing.
Once you get inside and you see what goes on here, this is groundbreaking stuff. This is affecting everyone across the world.
Without basic research, there would be no hope for my child. So we're very hopeful for the future for the first time.
Okay, so yeah, so just to conclude, RNA splicing is really emerging as a promising target for disease therapeutics. So there's already a couple approved drugs that target splicing and modulate splicing. There's some in clinical trials, and there's many in preclinical trials for many different diseases. And actually, this is an old slide. There's definitely more going on with looking at splicing and trying to modulate splicing for therapeutics for a number of different diseases. Okay, so I'll just sort of leave it there. I've gone way over time. I start talking about these things and I just start rambling on. So I'll stop sharing, see if there's any questions in the chat.
Yeah, don't worry about the
Any
time.
questions? Yeah. You start talking about these things and you
I know,
you
especially
get
when
into
it's
them
something
and
you're
then-
interested in, and so that was quite an emotional end to the talk. That I thought.
Yeah, no, it's really nice to see that the research into this has effects on people and can help people with certain diseases. And there are quite a few diseases where splicing is disrupted, where people and drugs are being developed to help them out.
Well, thinking about treatments, I was going to ask you if there was you know, there's no way anyone with any of these conditions could be cured. But obviously, that's looking at the SMA video of that little girl. That's the closest you're going to get to it, really.
Yeah, the SMA is just amazing. It's a really great story, and it was developed over years and years of how first they looked at how the splicing was disrupted, and then they looked to see if they could get that exon back in, and then they realized that if they went through all different cell models where they could add these oligos and see that it came back, and then when they did the trials were so promising. And I think it
makes you think that you could cure anything by splicing.
Yeah,
almost.
We can only hope. Yeah,
yeah.
I think it was good in that because it's very specific to the neurons and the spinal cord. They could get the drug right in there by doing these injections into the spinal cord, and it gets in there, and it can have an effect of stopping the progression of the disease.
Well, I think that was amazing. Anyway, it's amazing.
So yeah, a few questions. How are the splicing treatments administered? Is it via the blood? So no, with the SMA treatment, it's via an injection into the spinal cord. With some of the other ones, what they're trying to do. So the problem with some of these, it depends on the drug, really. So there are some chemical drugs that can influence splicing, and they can be given right through the blood. But if it's something like these DNA oligonucleotides, they are quite resistant in the blood, but not perfect in the blood. So it's not as easy. And then with a lot of these diseases, it's trying to target the right tissues and the right cells to change the splicing, maybe just in those cells. Does one mutation affect all the spliceosomes from Caroline? If it's a mutation in one of the spliceosome proteins, one of the spliceosome components, yes, we do think it affects all the spliceosomes, but we haven't proven that. So yes, if you maybe bring down, what we're thinking is if you bring down the levels by mutation of some of these spliceosome proteins, we're thinking that maybe only some of the spliceosomes may be affected and not all of them. So that's a good question. How young do they have to be for it to work, Helen? I think at least in that example there, that you had to wait for the SMA. I think the patients had to wait until they were 2 years old to get that, but it depends on what type of SMA that they have and how severe it is. So I think they can be treating earlier on, but I think there is a sort of an age that they have to get to before they can be treated. Any other questions? Trying to think what's coming up on there. Can you treat an adult with it? Yes, adults with SMA can be treated, and they can be treated, and it can halt it can't reverse it, but it can halt the symptoms that they have already, and they won't progress any further. How much does Spinraza cost? Okay, yeah, it's quite expensive. Yes, I don't know exactly how much it costs, but it is quite expensive.
Has anybody else got any more questions?
Hey Ray, it's Martin.
Hi.
I've heard about coronavirus vaccines, and
Yeah.
some are based on, is it RNA or mRNA?
Yeah, they're messenger RNA vaccines. Yeah, so the Pfizer one and the Moderna ones, they're messenger RNA. So exactly what I was talking about. They're the RNA that's used to make proteins. So yes, definitely.
And if you had a choice out of the vaccines, which one would you have?
I was just about to ask that because I get my vaccine on Saturday.
What are you going to get, Sarah?
I don't know, but I'm going to the same place that my partner had his done in early February, and he had the Pfizer one. And I think it sounds like the Pfizer one might be the better one. So I'm hoping it's that.
Yeah, it's yeah. There's obviously the mRNA ones, and they seem to have really good. They're working really well. And then yeah, I've seen in the news all this stuff about the AstraZeneca one, which is
Yeah,
a
I
DNA
really-
one.
I don't really buy into that. It's
Yeah, so who knows? I
not enough
haven't
people,
seen
is
any
it,
of it.
getting blood clots for it to be. I don't make the connection personally.
Yeah, it's still the
What?
the World Health Organization is still supporting it. But yeah, no, I guess the only the disadvantage of these mRNA ones is that they don't last as long because RNA is more is broken down easier than DNA. So they don't last as long, but you still get that burst of protein being made from the RNA, and it's long enough and strong enough to elicit the immune response. So that would only
But is that the reason the immune response wears off after a few months then?
yeah, well, the immune system then just goes down, but and it's usually why you then get the second dose of the mRNA one as well, just in case it was broken down too quickly and you didn't get enough protein being made from that mRNA vaccine.
Ah, that's interesting and topical. Thank you. Any more questions,
Yeah.
anybody? No more in the chat.